For today, I wanted to talk, as I've already said, about trajectories in what we call pseudotime. And I thought I'd do it this way. I give you a quick bird's eye view. So I prepared two kinds of material, one part which is on the biology and one part which is on the math behind it. And we'll start with a quick run through the biology part when we look into the math and when we go back to the biology file to have a closer look of what was happening there. And here, this one is the biology part. And what I've done there is I've taken our usual data set again that we had before. You remember this data from the neurogenesis in the adult mouse brain. You remember I told you there is this stem cell area next to the lateral ventricles where there's these neural stem cells that the Martin Vallalva group at DKZ is interested in. So they extracted these cells and their progeny and wanted to see how they go ahead. And we did this already before. Last time we did it, the UMAP looks different. I'm actually a bit surprised that it looks different today than last time. I'm not quite sure what I changed on my computer. But anyway, this is the UMAP we had this time, even though I loaded exactly the same data and ran here through my UMAP analysis. The best I can think about is that maybe the number of principal components was different last time. And just to get an overview of where we are in this data, I plot here the usual markers that we always use in this neurobiology to understand what we are. Aquaporine 4 is a marker of astrocytes. So for the non-biologists, which I think is nearly everybody today, except for you two, the aquaporine 4 astrocytes are those cells, those helper cells in the brain which supply the neurons with nutrition and do all kinds of other cleanup stuff. But interestingly enough, it seems that they are still quite close to the original neural stem cells, which were able to give rise to all the neural cell types, so neurons, astrocytes, and microglia, and whatever I've forgotten now, oligodendrocytes. That was the one I had forgotten. And so it seems that these astrocytes are quite easily to convert back into stem cells, and then are able again to give rise to all these 4 or 5 different cell types, and that's what we observe here in the subventricular zone. So here aquaporine 4 is a typical astrocyte marker, so these cells are all astrocytes, but also stem cells, because we consider the stem cells now as a special kind of astrocytes. And here they are dormant, and you see we have here this smooth trajectory of cells going from here towards here, which we interpret as these are cells which start here and they slowly develop to the state represented by this area in the state space. And because we see them slowly going through here, we imagine that, that this is a continuous process, and we just got a steady state snapshot of this where we see cells in all the different intermediate states along this journey from here to here, and this is why we see this continuous area. And to go on with our orientation, MKI67 is the standard marker for cell cycle. It's always upregulated when the cell goes into mitosis, so it's dividing. So it's used to see where the cells are dividing, and we see it here, and it's quite reassuring that it's a cycle, where we can really see the cells probably come here when they go through the cycle, and after they have one cycle, they again, as they were before, but one cell has become 2. Of course, the actual division was something we don't see here, how the number of cells increases going from here to here, except for maybe that the point cloud gets denser in state space. Double cortine is a marker for neurons and neuroblasts, so for immature neurons. We see these cells are so on the process on their way to becoming neurons. And GRIA1 is a marker for mature neurons, which are now ready to be brought to work. And as you can see, we have a jump from here to here, but this is only an apparent jump. That's, as I can show you by trying to open this sleepwalk part. Let's use this here again. So you remember this was this tool where I put it where I point my mouse to a cell, and then all the other cells are colored according to the Euclidean distance in this 20-dimensional PCA space. And so it's quite easy to see all these big blob of cell, they are all similar, while these little protrusions, they are quite different. So my guess is that these are sort of the normal, the astrocytes, which can also become stem cells, and these are specialized astrocells, which have already specialized enough that they are not stem cells. And these are then the cells which get activated, which are the activated stem cells. And when we go here into the cell cycle, and you see it goes around here. And one of the questions we always have about the topology is, you see these struts here, is this real or is this just an artifact of a 2D projection? Is there really different paths of getting from here to here? That's something we are currently trying to find out in this data set. Because the standard view in the literature says that these stem cells, in order to become mature neurons, they divide at least 2 or 3 times, giving rise to 4 or 8 or maybe 16 successor cells. So we have to go this cycle around at least 2 or 3 times. But here it also looks like you can go straight through. We can think later about, we will discuss later more about how we can actually explore such topology questions. But for the moment, let's keep it simple and just say we want to make a pseudo-time where we give each cell a number which gradually increases from 0 here to maybe 0.5 here up to here 0.7, and then we reach here 1. To justify that I consider these two connected, I go here to, where was it? To this little corner. And you see, if I go here, it seems that this cell here under my mouse seems to be quite closely connected to that ones here. So the way how I interpret this is that these neuroblasts, this is where all the cells which come out of a cell cycle, they all sort of accumulate, and we get a lot amount of immature neuroblasts. And now in the biology, you need to know that most of the cells, the neuroblasts that are produced, where they don't stay in the subventricular zone. They go on the migratory path forward to the nose and they replenish cells in the olfactory bulb. So most of the mature neurons don't stay where we are. And the neurons which naturally are there and have been all the time, we don't get them because we only get rather fresh cells because they used this sorting for cells expressing the GLAST marker in order to make sure that they only get cells which have been recently produced out of a stem cell. So we have only few neurons, and they are probably quite different because now once the neurons become neurons, they have to spread out and differentiate a lot. So there's probably a huge change in state when the cell progresses from here, this immature state to this mature state. And there's only few cells which do this huge jump, so we're missing that. We basically see this trajectory, and then cells, very few cells progress from here to here, and we just don't have enough cells to see anywhere along this intermediate thing. Another aspect is that this is maybe an artifact of a projection. As you can see, we have to jump from here to here. This connection here is very small, and we'll get later into how this UMAP is actually drawn. I wanted to make a lesson about this maybe in a week or two. And what we learn then is that the UMAP takes the neighborhood graph as input. You remember these 20 nearest neighbors, and because there's so little distance here, this cell probably considers mainly these as its neighbors and only few here, so that the neighborhood graph has maybe only 1 or 2 edges connection here. And this is not enough for the UMAP algorithm to realize that these two things should go together. So this is something you often have. These UMAPs can be wildly misleading, and this is why I like to use this tool that we made here in order to explore this. But what I now want to do today is just put a trajectory through this. So I want to fit a curve through a 20-dimensional principal component space that somehow starts here, goes through the middle of all this, and ends here. And here you see this is the result we will get. We will call a function simply called the principal component package to a principal curve package to calculate a principal curve along here. And for this, what I've done, I took all the cells which you now see highlighted in color. So all the cells in the lineage from 10 over 0, 3, 5, 1, 2, up to 7. When I tried it the first time, I also included the cells in 6 and 11. But if you look in the cell cycle actually changes the state quite drastically. So these cells here, they are very different from these cells. If I go on these cells, you see there is much less change here. So what I imagine is that in a way, the state space is, if these are the stem cells, we go to the intermediate amplifying cells and then to the neurons. And in our chosen metric, the change due to cell cycle is much larger than the rest. So going from stem cells to amplifying cells is a much closer distance in PCA space when the diameter of the cell cycle loop is. And then again, from that stage to the neuroblasts is also a much smaller distance, while the change in these neuroblasts is quite big. So if I now try to fit a curve which goes more or less through the middle, this curve will more or less be forced to move into this loop and end up here and won't find its way back.
But based on the assumption that there is a direct path.
Yes, in a way I did that. It's a bit cheating. What I really want the tool to do is to just find some closest connection from here to here, and then I later will project these cells onto the curve. But you're perfectly right, this is not quite clear whether this is the proper thing to do. And of course, at the moment, I just wanted to have a nice plot to demonstrate the fundamental method, and then how to deal with the cell cycle is still a bit an issue, and we can discuss this a bit later. So, yeah, now, so that's what I did. I calculated this, I took this pseudotime which goes through here, and the logic of this pseudotime is we want a curve, so a map which maps the unit interval into the 20-dimensional PCA space, so a map from unit interval to R20, and it just assigns this to a continuous curve starting here, going through here, and ending here. And I want to have it such that each cell, when I project it onto the curve, is as close as possible to a curve. And I define this in the following manner. I think I've put the formula on the other slot, so I'll show it to you in a second. But you can imagine what I intuitively want to get is I have this curve from my unit interval to my RD, where D is the PCA space dimension, and I have xi is the PCA coordinate of cell I, and ti is the position in the curve. So we want that we say for each x, let's write it properly. So let's say xi is D is the position of cell I in cell I in PCA space. And what we now want to find is we want to find a curve gamma such that if we project xi on gamma, we get the projection distance is short. So first I have to say what I mean by projection. So by projection, I mean I take this xi and find a point on the curve, gamma t, and I want to find the point on the curve which is closest to my xi. So I take this here and say I find the curve parameter which brings me closest to the point xi, which is obviously because this is a continuous curve perpendicular to it, so that this is xi and this is gamma of ti. If I make, and in that sense, I want now I want a curve such that all these projections get minimal in the sense that if I sum this up, over all xi minus gamma ti squared over I. This should be minimal in some sense. And I want to find the curve that minimizes that in a suitable sense. This is sort of the intuition. In the end, you will see that this principal curve algorithm of Hastie does not exactly do that, but is motivated by the desire to minimize this thing. So here now we have this curve. What I did next is when all the points which have not been involved in the curve fitting. So obviously I don't show you the curve here on this plot because you can imagine the curve runs in 20-dimensional space, and I don't know how to project the 20-dimensional curve in my UMAP because the UMAP projection is not a continuous map from the 20D to 2D. It's something weird. And because it's not continuous, and especially it's not clear how I can project an extra curve into it. So what I do instead is that I color each point by this pseudo-time value Ti, and then you can imagine the curve runs somewhere through the middle. What I can also do is that I color each point according to its distance to the curve, and then you can see the curve probably runs where the distance is minimal, so where we are here at the most yellowish. And what I've also done is all the points which I did not include in this minimization, I can still project them onto the curve and hence assign them a pseudo-time. I only have to be wary about whether pseudo-time has much interpretable meaning. But if I do this, then now all these points in the cell cycle also now get a pseudo-time. Even these points here get a pseudo-time, which are not part of a lineage. They are oligodendrocytes, and these little other cell population, which was our bycatch, they don't belong to it. We also get pseudo-times on them, but we will ignore them, but we keep this one here. And what I then now once I have brought this in this thing, what I can do next now is that I take any gene and plot the expression of this gene against the pseudo-time. And that's what I show you down here. Here I took one of these genes, SL1A3, GLAST. That's this gene that we use to pick out our cells when what they used in the lab in order to pick out which cells they actually want to include in the experiment, namely only those cells which express the protein produced by this gene. And what I've done here is each point is a cell. Specifically, it's a cell from the lineage, so from the colored cells here plus those here. So this is what I call the lineage, everything which is colored here plus these two clusters. And I took these, and each cell is one of these points. The x-axis is the pseudo-time, so this curve parameter, which tells us how far it is, and the y thing is our expression strength for this gene, again on this log scale, where we take the count divided by the total, add the pseudo counts, and then take the logarithm. One thing I've done here at the bottom is with 0. We see there's a lot of cells which have expression 0. And because it's so many of them, usually this would just be a line, but to make it more easy, how many points are on top of it, I've broadened the line a bit, simply by saying if it's 0, then replace the 0 with a random number between minus 0.2 and 0. And with that, we get now this plot here. And now I can do the next thing and ask, can I now draw a smooth line through this point cloud, which shows me how the expression typically differs from that. And this is what I've done here with this magenta line. As you can see, it goes here through the middle, and here it does something funny. It goes below and up again. And that's actually not a mistake, but this properly. Because remember that the way where this 0 is placed and how much distance we have here between the 0 and the 0.5, that's just an artifact of our normalization procedure, because we made this way of saying this is the decadic logarithm of the counts for the gene over the total counts times 10 to the minus 4 plus 1. And this scaling factor, 10 to the minus 4, it influences how much difference we have, basically how much this offset 1 here has against that, and this will cause this distance between the point cloud and the zero line to change depending on you put less or more on that. And of course, we don't really want that, because what we actually think is that it should be possible to fit a proper expression value, which is the proper average, including the zeros. And we'll talk later today about how to best do that. And this then takes into account that here the proportion of zeros among all the points is much higher than over there. And because of this, the curve goes down and up again, because here in this area, the proportion of zero is highest, while here it is less. And it actually takes into account this proportion of zeros. And we will achieve this afterwards by making not a least square fit, but a maximum Poisson likelihood fit, which will be the trick to get this right. But once I've done this, I can now do for each gene that I have such a curve, and then I can make a plot like this one here, where each row is a gene, and this here is the pseudotime starting from the astrocytes over the cycling cells to the neuroblasts and the neurons. And you can see how some genes peak early and some peak later. And here I've made this for 100 different genes, and you can see how this goes out and over. And this is the kind of way how we now get to something that from a biological point of view can be interpreted, because we can now see in which orders do the genes go up and down in order to, and so we can see the program that the cells execute by going through these different expression things. So our aim is to get to a plot like this. And in order to do that, of course, here is the code, but we'll need a bit of math in order to be able to do this. And for this now, so I will later go through the code here in detail, but for now, let's have a look at the details, and where I want to switch this around. Before I go to the question how we fit this pseudotime curve, I first want to talk about how we do this fitting a line through such a scatter plot. In general, this runs under the term of smoothing. You have a data cloud and you want to get a smooth curve through it. And I want to show you now 3 different techniques to do this smoothing. And then we will use this technique for both our use cases, one for fitting one gene against the pseudotime and the other for fitting the curve into PCA space. But so, how do we do that? Let me have a look at this thing here. So, in order to do this, what I've done here is I first started to make let's turn it this way around. I have here made a point cloud, and my aim is to make a smooth curve through this point cloud. Now, this should be considered a function of x mapped to y, because afterwards we have to distinguish between cases where we map from a parameter into a multidimensional space, and here we just map completely ordinary from one number to another. So this is an ordinary R to R function, which maps real numbers to real numbers. But it's not a function, because for each x values, we get here a whole spread of y values. And often, quite often, if you argue about such kind of things, it turns out to be very useful to have a data generation model in mind. So in practice, we will be presented with such a data cloud in some application and say, find a smooth curve through this. But and this data cloud is the starting point, but it helps to have a model in your mind of how such a data cloud might have been generated. So that has two advantages. One, you can simulate the generation of such a data and hence can produce example data on which you can test the algorithm you're developing. In the other idea, it's easier to reason about what your algorithm actually does if you have some story about how your data is generated. And then you can also argue about the fact whether your model of how the data is generated actually matches your real case. And in my experience, this is one of the core differences between physicists and mathematicians thinking. In physics, we do this a lot, that we always insist when we have data, we want to get a data generation model below so that we can say, are the residuals here normal or Poisson or whatever distributed? While in statistics and people like to do it the other way around, where they just look at the data, say, this looks normal or this looks gamma, so let's assume that. Here, of course, I know what it is because I have generated this example data myself, so I know how I made it, and I show you how I made it. I first produced a smooth curve, and I tried to find an example for a curve which has different challenges. A gradual ascent, when a very steep descent, when a rather horizontal thing and something like this, so that the smoothing problem is not too easy, that it's not something obvious like a sine curve and so on. And the way how I managed to do that is by using splines, and we will see how a spline works later. So for now, just consider this BS function that you see here as a magic way of turning these numbers into a smooth curve, and the magic will be revealed in half an hour. So I have this function which I call the true function because it's the underlying ground truth which we try to recover. And this true function maps the unit interval to these y values here. And when we assume that the data we are actually able to work with is not the true function because the truth is inaccessible, we only get a noisy variant of the truth, where we put around this thing where we add random numbers on that. And we first assume that we don't know the value of this curve at all data points, but only at 100 randomly selected x values. So I select 100 random values between 0 and 1 drawn from a uniform, and I calculated the true function at these 100 points, and then I add noise from a normal distribution with mean 0 and standard deviation 1/10. And if I plot this, I get this curve here, where you see the x values are drawn uniform at random, the y values are drawn from the curve plus a normally distributed noise. And that fact that our noise is normally distributed will be important later because we will use the least square criterion to recover it. And hence, we need to make this assumption that this is normally distributed noise. And later we will discuss how to do that if the noise is not normally but Poisson. Now, if I want to make a curve, the most primitive thing is I just bin the x values. I say, okay, I put them in 10 bins from 0 to 0.1, from 0.1 to 0.2, and for each bin I take the average. So that's what I've done here. I've binned my x. Sorry, where is my I've just put x value into a bin, and when I calculate the mean in each bin, and when I make this plot here, where I plot the mean x and y values against it, and I can recover my curve, but now this is sort of a staircase curve, or a very angled curve, and it's not smooth, and our aim was to get something smooth in, so it should at least be once or twice differentiable. And if I just connect these points, I get something continuous but not differentiable. So we want to do better. The next simple thing is kernel smoothing, which is probably also something you all have heard before. What I do there is, right here you can't see the plots, so let's quickly go over here. For kernel smoothing, what I say is that for each x value, I take a continuous gradient of x value, so I want to be able to get a value not only for discrete points like here, but for any arbitrary x value, even something between the data points. And I say the smooth function value at this x value x0 should be a weighted average of the y values in the vicinity. And this weighting I do such that I say those y values which are close to my target x value x0 should get a high weight, and those which are a bit further out should get a lower weight. And for this you use a kernel. And you just say the weight should be this kernel which is evaluated at the distance from the x0 thing. And kernels usually look like this. Here you see a couple of typical kernels. This might be the simplest kernel, the boxcar function, which is just constant between -1 and +1 and 0 outwards. So it essentially means for every x0 values, you take the average of all those y values which correspond to an x value, which is no further than 1 from it. And if you don't want to say the width of the window over which you average is 1, but something less or more, you of course have to divide the difference by some kernel width h, which I have. So this would be what you call a moving average, where you just go to a point and put a block around and say from -h to +h, I want to get this. But you might want to put more weight to the stuff in the middle than to the weight at the order to get more smoother transitions, because otherwise, if it wouldn't go down, once your average just goes over a new data point, you get a step. And in order to not get a step, you want your weight to smoothly increase. And a popular kernel is this one here, the tricube kernel. As you can see, it has a couple of nice properties. First, it's twice derivative. So you have a continuous first and second derivative everywhere, especially also here at this breakpoint where it, to the left, it's exactly 0, and when it goes out, it's sort of flat in the middle, so that it somehow has some internal smoothing out small differences. And yep. So typically when people choose a kernel, the first thing everybody thinks about, if it's not a simple moving average, is to say, let's take a Gaussian, but a Gaussian is annoying because it doesn't have finite support. It goes all the way to infinity. So you want something that looks like a Gaussian GAL, but has support only between -1 and +1. So you might come up with this tri-weight thing. If you can say, actually, I want it to be not as pointed, but more flat in the middle, when people get this tricube, which is sort of a standard smoothing kernel that everybody uses. And here you see it's called tricube because you take the cube of the kernel argument and then you cube it again. And you have some strange prefactor, which is just in order to make sure that the integral under the kernel is 1. But because I divide by the sum of the weights anyway to get my weighted average, I don't need this thing. So my tricube is just here, this function without the prefactor. And if I use this to smooth, I can now say, okay, take the point where I want it, calculate the tricube there, and calculate the weighted sum. And if I do this, I now run through 300 points between 0 and 1, enter each of these 300 points for x0, and then I get this red curve, which, as you can see, follows the original blue curve quite well, except for this point here, which is expected because we cannot follow such a steep smooth very well because we've chosen a kernel which is a bit flat on top. Maybe it would like a little bit better if we had used the tri-weight kernel instead of a tricube kernel. But we can also do something else, and that's called local regression. Before I go to local regression, let's quickly talk about this bandwidth h here. So here is what I have here, and here you can see my kernel, and of course I can change the bandwidth, so the width of the kernel, how wide the support of the kernel should be. If I put it much bigger, for example to 0.5, then you can see now the thing gets over smoothed. Or let's make 0.2, then you can, and now you can see the wider I make the kernel, the smoother it gets, the less the thing is able to follow these tiny little derivatives. But if I now conclude when I should make a small smoothing bandwidth, then obviously this here happens. And if I make the bandwidth too small, it will follow every little nook and cranny in here. And my aim is to choose a bandwidth which makes a good trade-off between being too wiggly and being too over-smoothed. So, and it's worth playing a bit with this, and I'll talk in a moment about a principled way to choosing smoothing, but that's how we do it. The next way to do local regression is the following. Let's have a look at this here, which is again our data points, and I put my x0 here somewhere at 0.35. And what I now want to do is, what I've now done here, I've drawn the points by the weight. So as you can see here, the size of the points given by the sex parameter is the tree cube weight. And this is now the points over which I average in case I want to get here the x0 point, which is here at 0.4, so here in the middle. And as you can see, for this 0.4 point, if I were to just average over points I have here, I would get somewhere here. And that's where our red smoothing curves is at 0.4. It's up here, but the blue curve is down here. And you can imagine if I draw a regression line through this thick thing, when the actual value for my blue curve, the regression line would not pass through the mean, but it would pass much further down because the regression line, of course, knows that it goes downwards. And that's what I do here. I fit a regression line just using standard ordinary least square fitting, the LM function, by saying fit y on x using the weights w, and then I get here these fitting parameters. Intercept 4, slope minus 0.9. I can draw this here, and I get my fitting line, and now you can see the value where the fitting line goes through my center point, 0.4, is a good deal below where it was before. And this is what I call local regression. Instead of just taking the average for each value x0, I make such a regression line, and then I take the value I take the y value where my regression line passes through my x0, rather than just the average. And if I do this, then actually here it hasn't changed that much, but sometimes it changes a lot. I want this to change a bit more. One thing I can do, instead of just fitting a regression line, I could also fit a polynomial of second order, so I fit a parabola to that thing. In R, I do this by changing, by adding to my LM function not only y tilde x, but I say y tilde x plus x squared. And I have to put here this x squared in such a capital Y to tell R I actually want to literally calculate x squared. Otherwise, it does some strange thing of thinking this is a mixed coefficient and I should mix something. So if I do it with that, then I get a Nash-Meisser fit. So here you see the red line is now follows now the blue line much better than before, and it manages to follow this steep part much better than we got it up here. And this is because we now used this local quadratic regression, where for each data point we made a parabola. And here you see in the R code how this is done. For a given point x0, I calculate the weights with my tree cube, then I calculate the fit by fitting y not only on x but also on x squared, and then I get two coefficients, an intercept, a slope, and the prefactor for the quadratic term, and I can calculate the fitted value for that. And now I go through my all my gradual x0 values and get this thing here. Next little trick in this fitting is before I drawn my x values from a uniform distribution, but now let's draw it from a non-uniform distribution. Here you see I came up with some strange mixture of two betas to draw my x, and if I do that, what I now, and I arranged it such that I get many x values here, and here's an area where we are sparse in x values. And you can imagine that's something we also have in our data where we said in the PCA space, sometimes we jump over a whole region, there's few cells in there. So this here is an area with sparse values, and if I do my quadratic regression that I've just done now, you'll see what happens is that here the fit gets very bad and jumpy. And this is, of course, because to the left, our kernel with its usual width of 0.1 covers many data points, and here it covers only few data points. So the bandwidth is sufficient here, maybe even a bit too generous, while here the bandwidth is too little. And you might want to choose the bandwidth adaptive. And the simple way to choose the bandwidth adaptive is simply whenever you want to smooth at a certain point x0, you say, let's find a bandwidth fitting for this place such that there will be 10 points under my kernel. And that's what I've done here. So you see, I changed my fitting function again. It's the same fitting function as before, where I calculate the weight, and then I do my quadratic fit, and I evaluate my quadratic fit at the point x0. But before I do this here. I calculate the distance of each data point from my fitting target point, x minus x0, and sort this. So I have them now sorted by distance. And then I ask, what is the 30th smallest distance? And obviously, if I choose the 30th smallest distance as the kernel weight, then the support of the kernel will cover exactly 30 data points. And this is called adaptive bandwidth smoothing, where you just choose the width of the size of the kernel or the width of the kernel such that it always covers a fixed number of points. In my case now 30. And if I do that, my fit looks better again. You see, now it's smoother. It's still maybe a little bit too wide here, but it sort of works. Yeah.
Go back a bit. When we use the weighted regression lines, shouldn't that, in the next plot, shouldn't that produce like a linear piecewise function?
No, because for each, and that's part of the trick here, for see here, I have this X grid, which is just 300 grid points. And for each grid point, I make a new regression. And you can make these grid points arbitrarily fine and make arbitrarily many regressions. So, of course, what I've plotted here is by necessity a piecewise linear function, because when you plot stuff on a computer, it always is. You evaluate your function at a grid of finitely many points and make lines in between, because that's how you do that. But typically you make enough lines that you have one evaluation per pixel, which is what I did here with 300. But in principle, the function is not linear, because even if I only move my x0 and epsilon to the right, the weights will have changed a little bit, and the regression line will change its coefficients a little bit, and the y value changes. And that's the whole beauty of this local regression idea, that even for you do a linear fit, you actually get a nonlinear output. And that's also why the quadratic fit is not about now allowing for getting even further from nonlinearity, but more for the function sensing curvature. So the reason why why my red curve wasn't quite able to follow the blue curve here, and with this with a linear regression. Which one was the linear? That is the linear where it, local regressions move. Now I think I've ran too many different. The reason why it couldn't, why the quadratic expression improved things, if you imagine I would now fit here a parabola in, the parabola would probably go like this, and see that we have a curvature here, and the slope decreases down there, and hence it can make the slope tighter here because it knows it becomes slower here. So More questions? Then let's go on. So this was our adaptive smoothing. As I said, we now use the fact for each x point we make a local regression, which is only valid for this very point, and even if we go an epsilon to the right, we make a new regression, and we can make this regression just fitting an arbitrary polynomial, and we always fit the polynomial such that the polynomial goes closest to the data points according to a weighted least square. So the vertical distance between the data points in the orange line squared and multiplied by the weights, that's what's minimized. And we'll see afterwards, I think I have it on the other slide, how we actually do this minimization. Who remembers how least square minimization is actually done mathematically? Because I also forgot and looked it up again. The nice thing is that it's not a gradient descent in the sense of that you try with coefficients and wiggle with them until they improve it, but you can find the optimum in one go by solving a linear equation system, and I have this afterwards because we'll need it for something related. So, but for now, this is our local regression, and as you've seen, I've written this local regression here in that manner. My local regression function works quite nice. It's not really difficult. R function, where was it? Here, you see, it's just sorting to find the 20th or 30th smallest nearest point, using this as the kernel bandwidth. Now giving this into the tree cube function, calculating the scale distance of each point to that. When we do the fit with the LM function, which solves this linear equation system, and when we use the fit to reconstruct the value at x naught, and we call this function for each grid value. This works actually reasonably fast, but in the end, you know, R is a bit slow when it runs, when it's about doing loops like this here. So it pays to have this implemented in C in case you need to fit many things. So in R, there's this package called logfit, which does all this in one go, or also LOES. LOES is a standard R function. Logfit is a package which adds a few extra things. So this LOES function does more or less the same what I've just explained so far. You just tell the LOES function, I want to fit the y values on the x values. I want to do each local fit with second degree polynomial, so a quadratic fit. And when you have this span parameter, which is for the adaptive bandwidth, you have to tell it how many points should be under the kernel, but it doesn't want a number of points, but it wants a percentage of all the points. That's why I divide my number 30 here by the length of all the points, because overall, you remember, I have 100 points, so I have to say span is 0.3. And if I do this, I now get here the red line. That's the line which came out of my own quadratic regression function. The orange line is the one that the logfit, that the LOES function from R does. So it seems the LOES function does the same that I've manually programmed before, because the orange and the red line are on top of each other. So what else do we need? As a fundamental rule in statistics, whenever you do any kind of inference, you want to know how precise this is. And what we've done here is an inference problem. As a reminder, the term inference means we have a sample and we want to infer information about the underlying population. In our case, the sample is our data points and the underlying population is the blue true curve plus its noise parameter. So our data generation process. Can we infer the parameters of a data generation process, so the real blue line from that? And of course we can. You see our red line gets close enough to the blue line, but we don't know how close. And the fundamental rule in statistics is an estimate is useless if you don't know how precise it is. So we need some way of knowing how good how well does our red line go to the blue line? Of course, here can be see this, but in the real world, you don't know the truth, because that's what you want to find out. So you can't check how close it is to the blue line. So you need inference techniques to find this out. And I want to show you two ways to find the precision of it. One thing is to use a standard least square diagnostic. So in ordinary least square, what you can do is, I go back to this one here, is I take here my regression line, and because I can calculate the precision, I can calculate a standard error for both the slope and the intercept, because I know the residual variance. So the variance of, remember, residuals are the vertical distance of a data point to the line, and the residual variance is the variance of this vertical distance. And we know that, and from this we can calculate the uncertainty of the slope and the thing, and from this the uncertainty of a fitted value for this red point. So for the red point's y calculation. And this way I get for each point in my local regression an uncertainty. And I don't go into the math now on doing this. You can read this up in every textbook on standard linear regression. But if I do that, then I get something like, if I put this here, where for each point I get this regression, and as you can, and here now I have used the lowest function as before, and it has this funny predict function. So if you use the lowest function from R and tell it predict my curve for all the values in my value grid, and also give me standard errors, then what it returns to you is here a list of fitted values and a list of standard errors. Now the first couple of values are NA, but in the middle they actually have a value. And I simply here, as you can see, I take my I take my fitted values and I add to the fitted values plus the standard error and minus the standard error in order to make this pink boxes. I multiply it by 1.96 because I want a 95% standard confidence interval. And you remember standard error means, according to the assumption of all our inference process, the estimated value scatters around the true value, normally distributed with this standard deviation, standard error as standard deviation. So we assume that and vice versa, we can say that the distance between the true and the estimated value is a random variable with this standard error. And the probability to be one standard deviation away from the mean, as you know, is 68%. Because that's just, if you take a normal distribution, that's the integral between -1 and +1 in a standard thing is 68%. And if you want 95%, you have to integrate from -1.96 to +1.96. And then you have an integral of exactly 95% under the bell curve. And this is why I multiply them with 1.95. Now I can say that the probability that the true value is within the band should be 95%. And I think that actually fits well. It seems that it's only in 5% the places where the blue line escapes from the red line. So this is one way of getting a standard error. Another way is because you have a lot of normality assumption. Here I know that the normality assumptions hold because I generated my data by adding normal noise. But in real life, you might have good reasons to believe that the data generation, that the noise which was added is not normal. And then you might distrust these regression diagnostics, which are all based on the assumption that the residuals are normally distributed. So there's an alternative to do that, which is called a cross-validation. There's two things, bootstrapping and cross-validation. Actually, and now I'm doing it backwards. Let's start with cross-validation, which is pretty simple. For each point, I calculate the regression value at this I calculate my fitted values now only at the x0 values, which actually correspond to data values. And to make the fit, I use all the data points except for that one. So each of these red points is now the fitted value by doing local regression over the black points, over all the black points under the kernel, except for the middle point. This is called leave one out cross-validation, or often abbreviated LOOCV. And if you do that, then you get here this red line where you can now see how far the red points are typically from the actual values. And we look at these differences, and that gives us a good estimate of a typical standard error. So this is one way, leaving out one data point and seeing how much this changes how close you get to the observed data point. If you try to predict this data point from all the other data points, and that gives you a good idea of the precision of your overall regression. So this leave one out cross-validation is one popular way to get an estimate of a standard error by, as you can see, I always fit it without that one. I took now the differences between the leave one out fitted values and the observed values and take the standard deviation of that and say, this might be a good estimate for the typical standard error. And the other thing is bootstrapping. Bootstrapping, an idea which has very generally used in statistics, goes back to Efron. Efron is the pioneer of non-parametric statistics, so he's the guy, he's actually still alive, some retired gray-haired guy in Stanford, who I'm very impressed by him because he's this guy, he sort of has influenced statistics twice. When he was a young grad student or a young professor, he was the guy who sort of was one of the pioneers of non-parametric statistics. And then 30 years later, when molecular biology came up and produced a lot of data and the first gene expression values came up back then, not with RNA-seq, with expression arrays, he was also the first one to realize that this is actually a job for statisticians and more or less pioneered this kind of biostatistics that we are doing there, which is something you see rarely, that somebody who's worked in one field his whole life and realizes actually this completely new field which paired up is where my field is useful and comes in that and then starts a new field when he's already about to retire, or maybe not quite. But anyway, so that's a guy who knows how to do proper research. I always wonder what to learn from him. But very long, at the very beginning of his career, he had this idea that he said, what do you do if you want to calculate a standard error from for a distribution which you don't know how it was produced? So typically, let's say you have data coming from some normal distribution and you want to know what's the standard error of this mean. You use your usual standard formula. You say, let's take the standard deviation and divide it by the square root of the length of this. And you get a good estimate for the standard error. If the size of the data set is small, you might use the student correction that you might know from the student t-test if you've ever looked into the math behind the student t-test. But in general, this idea relies a little bit on the idea that the errors are normal. And in case of the mean, the errors are actually typically normal because a mean is a sum of very many data points which when divided, and whenever we sum up many random variables, of course, the sum will be normally distributed according to central limit theorem. But there's many statistics that we might calculate that are not the sum of individual data points, and hence where we cannot assume central limit theorem. And what do we do then? And typically, the ideal case, if you really want to know the sample distribution of this mean, is the following. I simulate that. Here you see I calculate 100 values, calculate the mean. I again take 100 values from the same distribution and calculate the mean. And if I do this 1,000 times, then I get the sampling distribution of my mean. But I only can get the sampling distribution because I can get new samples from my underlying distribution. But in the real world, I don't have new samples. I cannot sample, I cannot get a fresh sample from the underlying distribution because I have only this one sample. And how could I now get something which is as close as possible as a fresh sample from the underlying distribution using only the sample that we have? And now Efron argues, in a way, we can draw a histogram from the sample that we have, smooth over this histogram and say this is now our estimate of the density of the underlying distribution, so let's draw from that. If we start smoothing, then we don't know if we've done this properly. So the actually most simplest possible way with the least assumptions is to draw width replacement from the underlying distribution. And this width replacement is the crucial part. So here I have my x. And if I now, and I might look here at the histogram of this distribution, and now if I now say sample x, replace equals true, that means take one of these 100 values at random, put it into our new sample, take another one and put into the new sample, but before taking a new one, place the old one back. Take a note of it and taking plot. In German, it's called, in English, you call it drawing with replacement. You have to put the stress on the re, because we don't replace something. We place it back into the urn. In German, you might know this is ziehen mit zurücklegen. So we replace our samples, and then we get a new sample from this. And the point of this new sample, it contains the same values as the old sample, but some values now will have been drawn multiple times, and others will not have drawn at all. And the funny thing about this thing is that it's as close as possible as we get of getting a completely fresh sample from the same data. Because due to the fact that we've drawn width replacement, we basically said each of these values has the probability 100th, and with this probability we have drawn it. And because each of our 100 values should be independent, we should always draw from the fresh whole sample and not from the sample the others already taken. That's the argument for drawing width replacement. And now Efron argues whenever you have any rule of calculating a statistics from a sample. So taking the mean, taking the upper quartile, taking whatever, you know, just to remember, the term statistic here means we have a sample, so a whole bunch of numbers, which we condense to one number by calculating a summarizing function like mean, quartile, whatever. This is what we call calculating a statistic. And whenever we want to get the standard error of such a statistic, and we don't want to make any distributional assumptions, what we have to do, the best we can do is to take new samples from this, like this here, and calculate the statistic again on this fresh sample. And if I do this now 100 times, then I get a reasonable sampling distribution of this statistic and can ask, how much does this statistic from this resampled thing, sorry, that was wrong, how much does it deviate from the true mean? So this is the mean of the original sample. This is the mean of the resampled samples. And I can ask how much do these deviate from the original sample, can calculate the standard deviation of these bootstrapped resampled things, and by this get a standard error. These Efron bootstrap standard errors have a tendency to have a downwards bias. They are always a little bit too small. Because, as you can imagine, if you would draw from the real distribution, in half of the cases you would get extreme values to the very left and to the very right, which are a bit further out to the original distribution, and that's of course something that cannot happen if you redraw from your existing sample. This is why you have to always keep in mind that Efron bootstrap samples are always underestimating the standard error a little bit, but they don't make any parametric assumption. So where the term parametric statisticians means we assume that we know the functional form of the underlying distribution. We know that whether it's normal or gamma or chi-squared or whatsoever. And if we don't want to make such an assumption, then this Efron bootstrap is what you do. To explain the name bootstrap, the Germans here might know the tale of Baron Munchausen, who always told these tall tales in these old stories. And one of his tall tales is that he said that he was riding through a forest, and his horse was jumping into a moor, and he was sinking down into this morass. And then he managed to pull himself out of this morass by pulling at his own hairs. In the English version of the story, Munchausen, or I think in the English version it's another hero, not Munchausen, but the hero there doesn't pull himself out by his own hair, but by his bootstraps. So by the straps that riding boots have to pull them on. And yeah, this is why in English you have these idioms: pull yourself out by your bootstraps, which means get something done without getting any outside help. And this is what we're doing here. We're using only the data we have and no further assumption. That's why Efron called this procedure bootstrapping. So after this short excursus into English language and statistical ideas, let's try this out. So what I've done here is I do bootstrapping by taking here the sample int function, which you remember I have 100 data points, and every time I call it, it draws another 100 numbers with replacement between 1 and 100. And then I call this my bootstrap sample BSS, and I use this as an index into my x and y values, so that those that I use with data points that are indicated here, like the data point 38 and 34, and some of the data points, for example, data point 2 appears 3 times, and so on. And each time I rerun my smoothing function and draw a line. I do this 100 times to get 100 lines. And now, as you can see, this is how my bootstrap sample looks like. Something went wrong. Yes, I've messed up my x when I tried this out. So let's do this again. And now you can see here, this is our bootstrap sample. And we can see quite nice how the bootstrap sample gives us a spread of how precise the estimate is. We can also see how it tends to underestimate the things a bit, like, for example, this problem here with the corner, the bootstrap simply can't notice. So, what's the next thing we do? As I said, we assume here that our noise is Gaussian. It's normally distributed. What we need is, but in our single cell data, we have Poisson data. You remember we assume that in each cell, a given gene has a certain concentration and abundance among all the samples, which is this fraction that we get here with K over S. But this fraction, of course, is a rough estimate, because in reality, we assume that K is follows a Poisson distribution around some parameter lambda, which tells us how strong this gene is expressed, times the number of stuff we get in total. So this K here is, so in a typical gene expression data, this data here is not a Gaussian, but it's a Poisson distribution. I want to simulate such data. So I first have decided that this time I take 300 instead of 100 data points. And again, I draw them uniformly between 0 and 1. And now the next thing is, because we have these different values S, I need some random values which I use as the size of the cells. And I've decided to just take a log normal distribution as this here. I take a log normal with a value of 1000.5, a mean around 1000 and 0.5, and in the end my log normal looks like this here. So you can see my cells have somehow between, what's this, 2.6 200, this might be 1,000, and this might be 5,000 reads. So this is a bit more or less what we also have in our real data set. And next, I use the true function, which we have before. But to make the true function a bit more interesting, I exponentiate it, because that's the same what we've done before always. So here you see I have taken the true function that I had before, but I exponentiate it, which makes everything a bit more pointed, even more pointed as it was before. And I call this true fraction now instead of true function, because it's the fraction of all the transcripts which fall onto our simulated gene. And I have adjusted here the value such that my fraction somewhere is between a 10,000th and a 100th. So, and as you can see, if you can now imagine that this is pseudotime, and that the gene with increasing pseudotime becomes stronger and stronger, and then it drops to nearly not expressed at all, and then it goes to this level. Now we can draw Poisson noise by taking each cell, and for each cell we now have a pseudotime that we got here from this X, and we have a size of a cell that we got from this S, and I can now say, let's calculate the fraction that the cell with pseudotime X should have, call this Q. And now I now draw from a Poisson distribution by saying, get me n values from the Poisson distribution and take this Q times S as the parameters, the rate parameters as a Poisson. And if I plot that, I get this one here. And now you see how and I use again the same projection we always use for in Seurat. I divide k over s, multiply by 10,000, add a pseudo count, and we again have this annoying gap between 0 and 1. And if I change the gap here, I get different gaps. And this is what I want to get rid of by making sure I use this pseudo count here only when plotting the stuff, but not when calculating. So I could, of course, now start and use a standard kernel smoother here on this value where I take let's first do the kernels. Sorry, this is what I wanted to do here. Down. The kernel smoother. So I first do a kernel smoother by doing it as follows. This was the kernel smoother I used before. I just take this width according to the kernel and take a weighted average using the weights under the kernel, and then making a moving average by move the kernel through. So this is now no longer local regression. We again just do simple kernel smoothing, where we weight each y value by the corresponding x values. And usually you would now say my y value is something like k over s, or logarithm of k. And I should put here for y, I should insert k over s. But as you can see here, I've done it differently. I moved the s to the denominator instead of the numerator. So this is my kernel smooth function, and here k is on the numerator and s is on the denominator. And if I draw this, it works reasonably well. I get my kernel smoothing here, and I get my lines which go up here. The blue line is again the true fraction. And as you see, I plotted this time the y-axis logarithmic. So this is why the true function looks like our original function again. And the red line is now the kernel smooth, where I again plotted the smoothed kernel value on the logarithmic axis. And it works reasonably well. It manages to deal well with all these zeros down here, which is non-obvious, because if I had simply put k over s here, I would have a lot of zero. Yeah, it might also have worked, but why did I put it that way? We had to have a k here on top and the s at the bottom. And to do this, to understand why this is appropriate, I do the following. I explain it the following. Here I have, in this case, I weight each y value with a weight w. But the y values, before I assumed that all the y values have equal precision because they all had the same standard, the same noise added to it. But now the noise depends on the size of the cells. Big cells have more noise, small cells have less noise, because we know that the variance of this k here is, of course, the same as the expectation, lambda of s. So I should put a higher weight on the bigger cells and a lower weight on the smaller cells. And now there's an old idea in statistics that if I want to calculate an average over a collection of random variables, and these random variables have different precision, then I should weight them in order to get maximum, then I should weight them such by their inverse variance. So the theorem says, if we have xi, which all have variances, which all have variances vi, and I want to somehow calculate the average of all the xi's, then I have to make a weighted average, weighting each vi by 1 over vi. And this will give me the average over all these things with the smallest variance. And you can simply do, if you want to know why, what you can do is that you just put here some unknown weights wi, and then ask what is the variance of this thing. And you say, how do I minimize this variance? In order to minimize this variance, you write this out. So you say this is the wi stays at the bottom here, and here you have this is vi wi. And when you yes, it's wi. So. And then you try to find what values do I have to use for the wi's to minimize this. And for this, you have to use a standard method with Lagrange multipliers. And if you just do the thing, which is a nice little exercise, then you realize that the wi's have to be 1 over vi. I think I put in the script the link to Wikipedia where they actually show you the calculation with the Lagrange multipliers. But if you want to try out whether you still remember how to work with Lagrange multipliers, you can try it yourself to see that, this is the maximum. But what I now do, if I now say I want to apply this to here, I say I want to make a weighted average of this ki over si, and for the weights, I now want to say the weight should be 1 over the variance. And I say the weight should be 1 over the variance, so they will be proportional to the si. And if you put this in, we say weights is this, is this, is this. And when you get here that the variance is proportional to 1 over si, so the weight should be proportional to si. When we set this in, and then the si's cancel on top and stay at the bottom, and this is why we now get this way that we actually put the weights in the numerator for the s and not in the denominator. So this is for simple kernel smoothing. The thing which we need to do for now we want to use our local regression smoothing. And before we used local regression with ordinary least square, but now we should use local regression assuming that we have a Poisson thing and we want to maximize the Poisson likelihood. And for this we use a generalization which is aptly called generalized linear models. I guess many of you know about this. Who knows GLMs? Okay, let's quickly go through the GLMs, and then we are through for today anyway. So I thought as an example, I use something like radioactive decay. So imagine you have some piece of radioactive material which has some given half-life of 10 hours or something like this, And here, as you can see, every couple of hours, we measure the radioactivity by putting a Geiger counter to it and count how many clicks we get within, say, a minute. At the beginning, we get 100 clicks in a minute, and when 5 hours later, we put our Geiger counter again to it, and it's only 60 clicks, and then it's only 40 clicks, and so on. So this is the number of clicks per minute, and this is the hours which have passed since the beginning. And of course, I've simulated my data to exponentially decay. And because here, I put here my simulation, and I said, the expected number of counts which you should get per minute is the 100 counts we have at the beginning times 2 to the minus t over 20. So 20 is the half-life. And the actual number of clicks that you do is not the expected number, but it's Poisson around that. So I use this as an input into a Poisson distribution, and this is how I get these expected number of clicks, which I have plotted here. And now I want to find out the half-life. And the simple way that you do in the physics beginners practical is you make a semi-logarithmic plot. So you make this plot again, and this time put the plot on the logarithmize the y-axis, and then of course our exponential decay becomes a straight line. But because of our Poisson noise, we get a lot of wiggliness here. And here's actually a couple of cases where we got only 0 clicks and they drop out from our logarithmic plot, which is annoying. So instead of simply fitting a line with normal regression into this thing, what's better would be to use a maximum likelihood fit. So we ask, we take an assumed value, we say our count should follow a Poisson distribution with alpha times e to a kappa the time. Kappa is the decay constant, alpha is the initial amplitude of the radioactivity. And of course, we know alpha should be 100 and the decay constant should be ln 2 over 20. Let's assume we don't know this. Then what we can now do is that we say, given 2 values for alpha and kappa, we can calculate what is the probability of observing the values that we got. So for each alpha and kappa, this here is the probability of observing the k values that we actually have in our data. dPros is in R the function which evaluates the probability mass function of the Poisson. And of course, we have to multiply all them to get the joint probability of making all our observations. But because multiplying a lot of small numbers quickly gets annoying, what I do instead is I tell the dProis function, please report the logarithm of the probability mass function, and I add up all these logarithms. So I get the sum, so I get the logarithm of the probability of observing the observed data as given these values alpha and kappa. And that's something we call a log likelihood. In general, the term likelihood is used if we calculate a probability and consider this probability not as a function of the data, but as a function of the parameters. So your usual Poisson function, you know, the probability of observing k counts from the Poisson probability with e to the kappa is, let's see, if e to the minus lambda, k lambda to the k over k factorial. I think that was the probability. And this thing, I can either say lambda is fixed and I consider it as a function of k, then I call it the probability of observing k counts given lambda. But if I have my fixed observation and say this observation is what I measured and counted, it won't change anymore. So k is constant. I want to find out lambda, then I can consider this as can say k is the constant and lambda is the function. It's a function argument, so I consider it as a function of lambda and not of k. And then if I consider it as a function of a parameter, I call it a likelihood rather than a probability. And the usefulness of a likelihood is that we can now find what is the fitting parameter by lambda by making a plot of this likelihood against different possible values of lambda. and inserting different values, always keeping the same fixed data, we will typically get something like this. And then we can say this is probably the real lambda of value because it's where the likelihood is maximum. And when optimizing such a likelihood, what you always do is you never optimize the actual likelihood, but you always optimize the log likelihood, which, as you can see here, is minus lambda plus k ln lambda minus ln k factorial. And this is why here the dPros argument has this argument log equals true. Instead of calculating this formula and taking the log, it calculates directly this formula, which gives you numerical precision. And that's important because we will add up many of these values because we add up this over all our 300 data points. Or how many ever we have. No, it's only 50 data points here. And if I do this, I can use the optim function, which just does gradient descent. So I tell it start with the estimates 15 and 0.1, and now try to find the likelihood function, which finds the values for alpha and kappa, which maximize what this likelihood function here returns to you. And as you can see, this thing then tries a couple of values. It does a simple optimization thing where it always just adds a small epsilon to either of the two parameters, sees whether the value goes up or down, and then after a value it reports back and says, I got the highest likelihood values for alpha is 92 and kappa is 0.03, which is quite nice because our real alpha was 100 and our real kappa was 0.3035. And the value of the likelihood here is 40. The optim function has this habit that it can only minimize, it cannot maximize. That's why I put a little minus here in front of my likelihood, because I want to maximize the likelihood. And as you can see, it had to make 117 tries to find this maximum, and then it decided that it found the optimum. Obviously, this is doing a little bit stupid, because if you really want to do this well, you do a gradient ascent or gradient descent. You get starting value, you calculate the derivative of a likelihood with respect to lambda and k, with respect to lambda and alpha, and then you always take a step into the derivative and you will get the optimum much bigger. In order to do this, I would have needed to give the optim function not only a function with a likelihood, but also a function with a derivative, which is not a big deal. I can easily take this thing and calculate the partial derivative with respect to lambda and the partial derivative with respect to alpha. And, but I was too lazy to put this out. So I just used the version of optim. If you don't give it a gradient, it falls back to a simple trial and error thing, which takes much longer, but here it still takes only a second. So this is how maximum likelihood estimation works. And now we can use it to set up a generalized linear model as follows. I say ki is the number of clicks to time point I, and I say this is a Poisson distribution over the expected number of clicks mu I. And then what you always do is you say this mu I we transfer onto a linear scale, here by taking the logarithm, when I get an eta I, which is just the log of this. And this eta I say, is now given by a linear model. So by some intercept plus some linear combination of parameters. And in our case, eta I is simply beta naught plus beta 1 our time. Why that? Because if you now undo the log here, then this turns into mu I is e to the beta naught times e to the beta 1t. So you can see that beta naught is the logarithm of what I called alpha before, and beta 1 is what I called kappa before. Yeah, beta naught is alpha and beta 1 is kappa, well, without the minus, because here I wrote a plus. And this way I can now fit here my optimum values to find the kappa, and this way I can now rephrase this whole optimization procedure that I've done here into what I call a generalized linear model. So have I done this before? Here I say regress
K on T. K on T means make a simple linear predictor which has only a constant term and a term proportional to T. So R always automatically adds a constant term whenever you write a formula like this. And assume that the likelihood is given by the Poisson, and the link between the Poisson's mean parameter and the prediction of this linear term is given by the log. This is what's meant here by a used Poisson family with link is log. And this is the GLM function, and the GLM function then does exactly the same as we've done before with this optimum and comes to the same conclusion, namely 92.03. Yes, you see again 92, but it gives it, of course, on the different scale. It gives us the betas, and I have to translate the betas in what we have before by exponentiating them. And this here minus 0.03 is already the same as we had before. The technical difference is my optim function, as I've just said, is stupid trial and error. The better thing would have been to calculate the gradient of this likelihood, which would have been easy. I was just too lazy, and then make a gradient descent where you always calculate the gradient, make a step in the direction of the gradient, calculate the function again to see whether it goes down, calculate the gradient again. And the only question is how big do the steps have to be, which is something we will also have to discuss at some point, because we will need a lot of gradient descent optimization in the later parts of this course. With GLMs does the whole thing a little bit more clever than a simple gradient descent. It uses something which is called iterated reweighted least square, where, oh yes, right, I was too lazy to write up how this works. And what I then did, as I asked ChatGPT, please write up how, please explain RLS fitting for GLMs. And here, as you can see, it made me a nice little explanation of what I've just told. You always have to specify the distribution of a response variable, in our case Poisson. You have to specify a linear predictor, where you have this design matrix X. In our case, the design matrix is just two columns, one column with all 1 for the intercept, and the other column contains the time values. And beta is our two things, and the link function, in our case, the logarithm. And then, as you can see, it explains us. We start with some random initial estimate, and what you then do is you make a Taylor expansion of this thing, of the likelihood. And if you do the Taylor expansion of the likelihood, that's what it hasn't explained here, you will notice that actually you don't need the real likelihood function. You don't need to know this. You only need to know the Taylor expansion, which tells you how the mean looks, which we have anyway, and how the second derivative looks, which is the variance. And if you do the math, you notice all the thing needs to know about the Poisson distribution is that the mean is equal to the parameter and the variance is also equal to the parameter. So it has this the way how the variance depends on the mean, and that's all you need to write down the Taylor expansion. Here it has written it slightly. And that way you linearize it, and then you have a normal least square problem, which you can solve with a normal least square algorithm, or specifically with a weighted least square that we've done before. And it calculates a beta estimate. It uses this to make a new thing, and here you have the iterated function. It's actually a very nice, elegant algorithm. I think I caught ChatGPT to make a mistake in here, so I have to double check this. But yeah, it even gave us here a little code. So I've talked a lot. It's nearly 12. How far did we get? I'm always getting slower than I actually want to be. I hope I don't bore you guys by being too slow and putting too much extra stuff into it. But yes, now we have this GLM going. So what I quickly, to finish it up, quickly tell you, what I've now done is I took my local quadratic regression function and adapted it for Poisson. by replacing what we had before here with our linear model with a GLM, still the same, with the weights and with a family. And one extra thing I needed, namely this offset, which is because I have this S in here. And so in my specific case, my linear predictor, log eta S, is of course some intercept plus something which depends on the on x and on x squared, and then because this s should be in here plus log s. And notice this log s is special because it doesn't have a coefficient in front of it. So it's not part of a real linear model. It's just something that gets added to the lambda s in the end. And that's called an offset in linear model terminology. A constant value that is dependent on the data and just needs to be ordered. And luckily the GLM function has this offset parameter that you can feed in such an offset. And yep, and with that case I get this back. And then, as you can now see, this works really nicely. My red curve, my fitted curve, follows the original blue curve quite well. And especially, it does a great job of here navigating this fact how the proportion of non-zero and zero values slightly goes towards higher values and actually does this. And that's the real advantage of using Poisson regression for this kind of fitting, that we can actually really make use of these zeros and how we influence the thing. Yep, I didn't get to spline regression. That's probably not so important anyway. I needed it for other stuff. That's why I put it into a lot more detail. But we'll talk a bit more about this, and then we'll talk about how to use all this to make a trajectory. To give you the heads, it's very simple to now actually do the trajectory. We just consider each individual coordinate in our 20-dimensional PCA space as one y value, which we regress against pseudotime using this linear regression. And then we do an iteration, and then magically this comes out. So, that was all I wanted to tell to you for today. Yeah, this was one of these typical things where you have a lot of computer science, a lot of programming. I jumped a bit over the code because the math is of course simple. Just doing a little bit of regression. We didn't go beyond quadratic formulae, but it's nice how you can easily implement that and go with along. So if you want to learn a bit of R, go through the details of the codes. And in the coming lectures, hopefully, we will graduate beyond just doing quadratic equations and doing something more sophisticated. Especially, I want to get a little bit into machine learning and neural networks eventually. Good. Then that's it for today, and see you, hopefully most of you, on Monday.